Diabetes is an entire group of diseases characterized by excessive blood sugar or high glucose levels in the blood. There are actually several different types of diabetes. Type 1, which historically has been called juvenile or insulin dependent. Type 2, which used to be called adult onset or insulin independent. And then there are other groups like type 1.5, gestational diabetes, type 3. Most forms of diabetes are either type 1, about 5 to 10% of total cases, or type 2.
She was 38 pounds when she was diagnosed, and she was 6 years old. In 2 years, she's gained almost 20 pounds. She was in the 10th percentile for height and weight. Now she's in the 50th percentile. She's right where she should be. Normal range for blood sugar is 80 to 120 for all of us non-diabetic type people. So if you know 80's your low range, she's been 36, she's been 32, and it's scary. Her tummy hurts, she doesn't feel good. Probably the scariest thing we had was when she came to me at our house one day and she said, "Mommy, I think I'm low. I can't really see that good."
We had family members that had type 2, but not anyone that young. And sure enough, on that night, we took him to the hospital, to the emergency room, and actually, when we brought him in, the nurses literally cried because he looked like a cadaver. He was very frail, and he still just kept apologizing to us about he was sorry. We didn't know what he was sorry for. Well, of course, the diabetes had an effect on his personality, but thank God to the hospital, they brought him through, and 21 years later, he is a grown man, a full-time student, full-time worker.
I would just say technology in general has changed the way you manage and you live with diabetes today versus 26 years ago. 26 years ago, I was excited if I could find a Tab. Does anybody remember Tab versus Diet Coke, right? It was the first sugar-free soft drink. And for a diabetic, that was just amazing. There was no Splenda. There was really, saccharin was not that prevalent even 26 years ago. And testing was almost impossible. It was very expensive to get a blood glucose home system that you could test your blood sugar with.
Type 1 diabetes historically has been called juvenile diabetes because it most often strikes among children and among young adults. That description is changing for a couple of reasons. We're beginning to see more individuals in their 20s and 30s developing symptoms of type 1 diabetes, and we're seeing more and more children developing type 2 diabetes, fueled probably in part by the rise in obesity among children.
I'm Ron Poteet. I'm area president for Regions Bank in North Alabama, and I am type 2 diabetic. About a year ago, as a result of a routine physical, my doctor said, "Hey, come in, I want to talk to you about something." And he said, "You've got it." And I said, "Well, what have I got?" He said, "Well, you're type 2 diabetic," which was not a complete shock to me because I have a family history. And so my grandmother, my mother, and now I kind of crossed that threshold. And so it was a sobering moment for me to say, okay, I've kind of crossed over into to this situation. And so it's made some pretty profound changes for me in the way I eat, in the way I exercise, and the choices that I make on a daily basis to help me manage through this. And so, but the byproduct of those changed choices are as I'm healthier, my weight has gone down, my blood sugar is under that threshold now, and so it's something that I intend on managing going forward. It's not something I don't think I can sustain. It's a different way of life for me.
The technological advances in genetics and genomics are amazing. 20 years ago, when we first started learning how to look at the human genome, and we developed, in the Human Genome Project, developed the full DNA sequence, or the entire genetic makeup of one person, it cost over $2.5 billion to do that, and scientists from all over the world collaborated to work on this. It was a huge, huge project. By the time we finished that project in 2003, we could sequence a second human genome, or figure out the entire genetic makeup of a second person for about $60 million. That's still way, way too expensive. And so the technologies have taken huge leaps so that we can now do this for about $6,000, and soon we'll be able to do it for less than $1,000. So HudsonAlpha scientists are using these tools of genomics to look at a number of human diseases, as well as to look at other organisms that are economically important. In the human, we're working on a variety of cancers, we're working on brain diseases, and importantly, we're working on a class of diseases that have to do with metabolism, like diabetes, type 2 diabetes, obesity, and related disorders.
Ironically, one of the best clues to understand common diseases is to study rare conditions. And one of the best examples of this is a condition that's called lipodystrophy, where children that are born with this condition have almost no adipose tissue at all, but they actually develop very severe diabetes. And the approach of genetics and genomics to that problem has been tremendously useful in understanding the principles that govern how the body produces fat, how it uses fat, and how it responds to fat. So lipodystrophy is a great example of how studying rare conditions, experiments of nature, tells us about principles that can be applied to common diseases that affect all of us. Experiments of nature are not just limited to humans. In fact, most experiments of nature are present in animals, and one of the best examples of this is the laboratory mouse that was used to discover a new hormone that's critical in determining how fat is produced and how the body responds to fat. And that new hormone is called leptin, and it turns out that there are rare mice that lack leptin and that lack the leptin receptor. And scientists didn't know that. What they saw was these fat mice, and somebody said, boy, these fat mice are really unusual. I wonder if we can figure out why they're fat. And pursuing that led them to discover this new hormone leptin and the receptor for leptin, and that's led to remarkable new insights in thinking about how to develop new treatments for diabetes and obesity in humans.
In both type 1 and type 2 diabetes, there are clear genetic and environmental risk factors. There are a number of genetic and environmental factors. We have a better handle on the genetics for type 1 diabetes, but there are still missing pieces in both. And we don't have a strong handle on the environmental factors. There are a number of longitudinal studies, so that means we start with a group of patients when they're newborns and we follow them for 10 or 15 years and identify who develops diabetes, in the case of type 1 diabetes. We look at all the things that they've been exposed to, and that's the best type of study to give us a sense of what are the real environmental risks. Those have been going on for several years, and I think in the next 3 to 5 years, we're going to get some real answers about environment.
At HudsonAlpha, we believe that genomics is the tool that will empower personal health. It will allow each of us to be responsible for our own well-being. I find that possibility truly intoxicating. If you are equally excited by the power of genomics to empower your health, the health of your children and your grandchildren, join me and give generously to HudsonAlpha.